Now we are going to read chapter number 6 of 11th NCERT Biology, that is Anatomy of Flowering Plant. You can very easily see the structural similarities and variation in the external morphology of large living organisms, both plants and animals. Similarly, if we are to study the internal structure and also find several similarities and differences. This chapter introduces you to internal structure and functional organization of higher plants. Study of internal structure of plant is called anatomy. Plant have cell as the basic unit. Cell are organized into tissue and in turn the tissue the organized into organ. Different organ in plant show difference in their internal structure. Within an angiosperm, the monocot and dicot are also seen to be very anatomically different. Internal structure also show adaptation to diverse environments. 6.1 The tissue. A tissue is a group of cells having common origin and usually perform common functions. A plant is made up of different kind of tissue. Tissue are classified into two main groups, namely meristematic and permanent tissue, based on whether cell being form a new capable to dividing or not. 6.1.1 Meristematic tissue. Growth in plant is largely restricted to specialized region of active cell division called meristem. Plants have different kind of meristem. The meristem which occur at tip of root and shoot and produce primary tissue are called apical meristem. Root apical meristem occupies the tip of a root, while shoot apical meristem occupy the distance most region of the stem axis. During the formation of leaves and elongation of stem, some cell left behind from the shoot apical meristem constitute the axillary bud. Such buds are present in axil of leaves and are capable of forming a branch of flower. The meristem which occur between mature tissue is known as intercalary meristem. They are in grass and regenerate part move by grazing behavior. Both apical meristem and intercalary meristem are primary meristem because they appear early in their life of plant and contribute to formation of primary plant body. Meristem that occur in the mature regions of root and shoot of many plants, particularly those that produce woody axis and appear later than primary meristem, is called secondary or lateral meristem. They are cylindrical meristem, fascicular vesicular cambium, interfascicular cambium and core cambium are examples of lateral meristem. These are responsible for producing the secondary tissue. Following division of the cell, in both primary as well as secondary meristem, the newly formed cell become structurally and functionally specialized and lose the ability to divide. Such a cell are termed permanent or mature cell and constitute permanent tissue. During the formation of primary plant body, specialized region of apical meristem produce dermal tissue, ground tissue and vesicular tissue. 6.1.2 Permanent tissue. The cells of the permanent tissue do not generally divide further. Permanent tissue having all cells similar in structure and function are called simple tissue. Permanent tissue having many different types of cells are called complex tissue. 6.1.2.1 Simple tissue. A simple tissue is made up of only one type of cells. The various simple tissue in plant are parenchyma, collenchyma and sclerenchyma. Parenchyma forms major component within organs. The cells of the parenchyma are generally isodiametric. They may be spherical, oval, round, polygonal or elongated in shape. Their walls are thin and made up of cellulose. They may be either be closely packed or having small intercellular spaces. The parenchyma perform various functions like photosynthesis, storage, secretion. The collenchyma. Collenchyma occurs in layer below the epidermis in most of dicotyledonous plant. It is found in either as a homogeneous layer or in patches. It consists of cells which are much thickened at the corner due to deposition of cellulose, hemicellulose and pectin. Collenchyma cell may be oval, spherical or polygonal and often contain chloroplast. These cells assimilate food when they contain chloroplast. Intercellular spaces are absent. They provide mechanical support to growing part of plant such as the young stem and petiole of leaf. Sclerenchyma consists of long narrow cell with thick and lignated cell wall. having few or numerous pit. They are usually dead and without protoplast. On the basis of variation in the form, structure, origin, and development, sclerenchyma may be either a fiber or scleroid. The fiber are thick wall, elongated and pointed cell. Generally occurs in group. In various parts of plant, the scleroid are spherical, oval, and cylindrical, highly thick, dead cell with very narrow cavities. They are commonly found in fruit wall of nuts, pulp, fruit like guava, pear, and sapota, seed coat of legume, and leaves of tea. Sclerenchyma provides mechanical support to organs. 6.1.2.2 Complex tissue. The complex tissue are made up of more than one type of cells, and all these work together as a unit. Xylem and phloem consist of the complex tissue in plant. Xylem functions as conducting tissue for water and mineral from root to stem and leaves. It also provides mechanical strength to plant parts. It composed of four different kind of elements, namely tracheids, vessel, xylem fiber, and xylem parenchyma. Gymnosperm lack vessel in their xylem. Tracheids are elongated or tube-like cell with thick and lignified cell wall and tapering ends. These are dead and without protoplasm. The inner layer of cell wall have thickened which vary in form. In flowering plant, tracheids and vessels are the main water transport element. Vessels is a long cylindrical tube-like structure made up of many cells called vessel members, each with lignified wall and large central cavity. The vessel cell are also devoid of protoplasm. Vessel members are interconnected through perforation in their common wall. The presence of vessel is characteristic feature of angiosperm. Xylem fiber is highly thickened wall and obligated central lumen. These may either be separate or aseptate. Xylem parenchyma cells are living and thin-walled, and their cell wall are made up of cellulose. They store food material in form of starch or fat and other substance like tannins. The radial conduction of water takes place by ray parenchymatous cell. Primary xylem is of two types, protoxylem and metaxylem. The first form primary xylem element are called protoxylem, and later form primary xylem is called metaxylem. In stem, the protoxylem lies towards the center and the metaxylem lies towards the periphery of organ. This type of primary xylem is called endarch. In root, protoxylem lies toward periphery and metaxylem lies toward the center. Such arrangement of primary xylem is called exarch. Phloem transport food material usually from leaves to other part of plants. Phloem in angiosperm is composed of sieve tube element, companion cell, phloem parenchyma, and phloem fiber. Gymnosperm have albuminous cell and sieve cell. They lack sieve tube and companion cell. Sieve tube element are also long, tube-like structure arranged longitudinally and are associated with companion cell. Their end wall are perforated in a sieve-like manner to form sieve plate. A mature sieve element possesses a peripheral cytoplasm and large vacuole but lack of nucleus. The functions of sieve tube are controlled by the nucleus of companion cell. The companion cell specialized parenchymatous cell which are closely associated with sieve tube element. The sieve tube element and companion cell are connected by pit field present between their common longitudinal wall. The companion cell help in maintaining the pressure gradient in sieve tube. Phloem parenchyma is made up of elongated, tapering cylindrical cells which have dense cytoplasm and nucleus. The cell wall is composed of cellulose and has pits through which plasmodesmata connection exists between the cells. The phloem parenchyma store food material and other substances like resins, latex, and mucilage. Phloem parenchyma is absent in most of the monocotyledons. Phloem fiber are made up of sclerenchyma cell. These are generally absent in primary phloem but are found in secondary phloem. These are much elongated, unbranched, and have pointed needle-like apices. The cell wall of phloem fiber is quite thick At maturity, these fibers lose their protoplasm and become dead. Phloem fiber of jute, flax, and hemp are used commercially. The first form, primary phloem, consists of narrow sieve tube and it referred to as protophlloem, and the later form, phloem, has bigger sieve tube and is referred as metaphloem. 6.2 The tissue system. We were discussing type of tissue based on the typical types of cell present. Let us now consider how tissue vary depending on their location in plant body. Their structure and function will be depend on location. On the basis of their structure and location, there are three types of tissue system. These are epidermal tissue system, ground tissue system or fundamental tissue system, and vascular or conducting tissue system. 6.2.1 Epidermal tissue system. Epidermal tissue system form the outermost covering of whole plant body and comprises epidermal cell, stomata and epidermal appendages, the trichomes and hairs. The epidermis is outermost layer of the primary plant body is made up of elongated, compactly arranged cell which form continuous layer. Epidermis is usually single layered epidermal cell or parenchymatous with small amount of cytoplasm, lining of cell wall and large vacuole. Outside the epidermis is often covering with waxy thick layer called cuticle, which prevent loss of water. Cuticle is absent in roots. Stomata are structure present in epidermis of leaves. Stomata regulate process of transpiration and gaseous exchange. Each stoma is composed of bean-shaped cell called guard cell, which enclose stomatal pore. In grasses, the guard cell are dumbbell shaped. The outer wall of guard cell are thin and the inner wall are highly thick. The guard cell possess chloroplast and regulate opening and closing of stomata. Sometimes few epidermal cell is vicinity of guard cell become specialized in their shape and size and are known as subsidiary cells. The stomal aperture, guard cell and surrounding subsidiary cell are together called stomal aperture. The cell of epidermis bear number of hair. The root hair are unicellular elongated of the epidermal cell and help absorb water and mineral from the soil. On the stem, the epidermal hair called trichomes. The trichomes in the shoot system are usually multicellular. They may be branched or unbranched and soft or stiff. They may even be secondary. The trichomes help in preventing water loss due to transpiration. 6.2.2 Ground tissue system. All tissue except epidermis and vascular bundle consist the ground tissue. It consists of simple tissues such as parenchyma, collenchyma and sclerenchyma. Parenchyma cells are usually present in cortex, pericycle, pith and medullary ray. In primary system, in primary stem and root in the leaves, the ground tissue consists of thin wall chloroplast containing cell and is called mesophyll. 6.2.3 The vascular tissue system. The vascular tissue system consists of complex tissue, the phloem and the xylem. The xylem and phloem together consist of vascular bundle. In dicotyledon stem, cambium is present between phloem and xylem. Such a vascular bundle because of presence of cambium possesses the ability to form secondary xylem and phloem tissue, hence are called open vascular bundle. In the monocotyledon, the vascular bundle have no cambium present in them. Hence, since they do not form secondary tissue, they are referred to as closed. When xylem and phloem within a vascular bundle are arranged in an alternate manner along the different radii, the arrangement is called radial, such as in roots. In conjoint type vascular bundle, the xylem and phloem are jointly situated along the same radius of vascular bundle. Such vascular bundle are commonly in stem and leaves. The conjoined vascular bundle are usually have the phloem located only on outer side of xylem. 6.3 Anatomy of dicotyledonous and monocotyledonous plant. For better understanding of tissue organization of root, stem and leaves, it is convenient to study transverse section of mature zones of these organs. 6.3.1 Dicotyledonous root. Look at the figure 6.6. It shows the transverse section of sunflower root. The internal tissue organization is as follows. The outermost layer is epiblema. Many of the cells of epiblema protrude in the form of unicellular root hair. The cortex consists of several layers of thin-walled parenchymatous cell. Within intercellular spaces, the innermost layer of cortex is called endodermis. It comprises a single layer of barrel-shaped cells without any intercellular spaces. The tangential as well as radial wall of the endodermal cells have deposition of water-impermeable waxy material suberin in the form of Casparian strips. Next to epidermis lies a few layers of thick-walled parenchymatous cell referred to as pericycle. Initiation of lateral root and vascular cambium during the secondary growth takes place in this cell. The pith is smaller or inconspicuous. The parenchymatous cell which lies between the xylem and phloem are called conjunctive tissue. There are usually 2 to 4 xylem and phloem patches. Later, a cambium ring develops between the xylem and phloem. All tissue on inner side of the endodermis such as pericycle, vascular bundle and pith consist of steel. 6.3.2 Monocotyledonous root. The anatomy of monocotyledonous root is similar to dicot root in many respects. It has epidermis, cortex, endodermis, pericycle, vascular bundle and pith. As compared to dicot root, which have fewer xylem bundle, there are usually more than 6 xylem bundle in monocot root. Pith is large and well developed. Monocotyledonous root do not undergo any secondary growth. 6.3.3 Dicotyledonous stem. The transverse section of young dicotyledonous stem shown that epidermis is the outermost protective layer of stem covered with thin layer of cuticle. It may bear trichomes and few stomata. The cell arranged in multiple layers between epidermis and pericycle consist of the cortex. It consists of 3 subzones. The outer hypodermis consists of few layers of collenchymatous cell just below the epidermis, which provide mechanical strength to young stem. Cortical layer below hypodermis consists of rounded thin-walled parenchymatous cell, which conspicuous intercellular spaces. The innermost layer of the cortex is called endodermis. The cells of endodermis are rich in starch grain, and the layer is also referred to as starch-sheath pericycle. Is present on the inner side of endodermis and above the phloem in form of semilunar patches of sclerenchyma. In between the vascular bundle, there are few layers of radially placed parenchymatous cell, which constitute medullary rays. A large number of vascular bundles are arranged in a ring. The ring arrangement of vascular bundle is characteristic of dicot stem. Each vascular bundle is conjoined, open, and with enlarged protoxylem. A large number of rounded parenchymatous cells with large intercellular spaces which occupy the central portion of the stem consist of the pith. 6.3.4 Monocotyledonous stem. The monocotyledonous stem has sclerenchymatous hypodermis, a large number of scattered vascular bundles, each surrounded by sclerenchymatous bundle sheath, and a large conspicuous parenchymatous ground tissue. Vascular bundles are conjoined and closed. Peripheral vascular bundles are generally smaller than central located one. The phloem parenchyma is absent and water containing cavities are present within the vascular bundles. 6.3.5 Dorsiventral leaf. The vertical dissection of dorsiventral leaf, the lamina shows 3 main parts, namely epidermis, mesophyll, and vascular system. The epidermis, which covers the both upper surface and lower surface of the leaf, has conspicuous cuticle. The abaxial epidermis generally bears more stomata than adaxial epidermis. The latter may even lack stomata. The tissue between upper and lower epidermis is called mesophyll. Mesophyll, which possess chloroplast and carry photosynthesis, is made up of parenchyma. It has 2 types of cell, the palisade parenchyma and spongy parenchyma. The adaxially placed palisade parenchyma is made up of elongated cells, which are arranged vertically and parallel to each other. The oval and round and loosely arranged spongy parenchyma is situated below the palisade cell and extend to lower epidermis. There are numerous large space and air cavity between these cells. Vascular system include vascular bundle, which can be seen in veins and midrib. The size of vascular bundle are depend on the size of vein. The vein vary in thickness in reticulate venation of dicotyledons. The vascular bundle are surrounded by layer of thick wall bundle sheath cell. Look at figure 6.8 and find the position of xylem in vascular bundle. 6.3.6 Isobilateral leaf. The anatomy of isobilateral leaf is similar to dorsiventral leaf in many ways. It shows following characteristics different. In an isobilateral leaf, the stomata are present on both surfaces of epidermis, and the mesophyll is not differentiated into palisade and spongy parenchyma. In grasses, certain adaxial epidermis cells along the vein modify themselves into large empty colorless cells. These are called bulliform cells. When the bulliform cells in the leaves have absorbed water and are turgid, the leaf surface is exposed. Then they are flaccid due to water stress. They make the leaves curled inwards to minimize the water loss. The parallel venation in monocot leaves is reflected in near similar size of vascular bundle, except in main vein, as seen in vertical section of leaves. 6.4 Secondary growth. Root and stem in length with the help of apical meristem is called primary growth. Apart from primary growth, most dicotyledonous plant exhibit increase in girth. This increase is called secondary growth. The tissue involved in secondary growth are the two lateral meristem, vascular cambium and cork cambium. 6.4.1 Vascular cambium. The meristematic layer that is responsible for cutting of vascular tissue, xylem and phloem is called vascular cambium. In young stem, it is present in patches as a single layer between xylem and phloem. Later, it forms complete ring. 6.4.1.1 Formation of cambial ring. In dicot stem, the cell of cambium present between primary xylem and primary phloem is intrafascicular cambium. The cell of medullary rays adjoining this fascicular cambium becomes meristematic and form interfascicular cambium. Thus continues a ring of cambium form. 6.4.1.2 Activity of cambial ring. The cambial ring becomes active and begins to cut off new cell both toward the inner and outer side. The cell cut off toward pith mature into secondary xylem. This cell cut off towards periphery mature into secondary phloem. The cambium is generally more active on inner side than outer. As a result, amount of secondary xylem produces more than secondary phloem. As soon form a compact mass, the primary and secondary phloem get gradually crushed due to continued formation of accumulative of secondary xylem. The primary xylem, however, remains more or less intact in or out around the center. At some places, the cambium forms a narrow band of parenchyma which passes through the secondary xylem and the secondary phloem in the radial direction. These are secondary medullary rays. 6.4.1.3 Springwood and atomwood. The activity of cambium is under control of many physiological and environmental factors. In temperate region, the climatic conditions are not uniform through the year. In spring season, cambium is very active and produces large number of xylary elements having vessels with wider cavities. The wood formed during this season is called springwood or early wood. In winter, the cambium is less active and forms fewer xylem elements that have narrow vessel, and this wood is called autumn wood or late wood. The spring wood is lighter in color and has lower density, whereas the autumn wood is darker and higher density. The two kinds of wood that appears as alternate concentric ring consist of an annual ring. Annual rings seen in a cut stem give an estimate of the age of the tree. 6.4.1.4 Heartwood and sapwood. In old tree, the greater part of secondary xylem is dark brown due to deposition of organic compound like tannins, resin, oil, gum, aromatic substance and essential oil in central or innermost layer of the stem. This substance make it hard, durable and resist to attack of microorganism and insect. This region comprises dead element with highly lignified wall is called heartwood. The heartwood does not conduct water but gives mechanical support to stem. Peripheral region of the secondary xylem is lighter in color and known as sapwood. It is involved in conduction of water and mineral from root to leaf. 6.4.2 Cork cambium. As the stem continues to increase in girth due to activity of vascular cambium, the outer cortical and epidermal layer get broken, need to be replaced to provide new protective cell layer. Hence, sooner or later, another meristem tissue called cork cambium or phelogen develops. Usually in the cortex region, phelogen is couple of layer thick. It is made of narrow, thin wall and nearly rectangular cell. Phelogen cut up cell on both sides. The outer cell differentiate into cork or phloem, while the inner cell differentiate into secondary cortex or phloem. The cork is impervious to water due to suberin deposition in cell wall. The cell of secondary cortex are parenchymatous. Phelogen, phloem, and phloem are collectively known as peridome. Due to activity of cork cambium, pressure build up on the remaining layer peripheral to phelogen. Ultimately, this layer die and slough off. Bark is a non-technical term that refers to all tissue exterior to vascular cambium. Therefore, including secondary phloem, bark refers to a number of tissue types, peripheral and secondary phloem. The bark that is formed early in season is called early or soft bark. Towards the end of the season, late or hard bark is formed. Name of the various kind of cell layer which constitute of bark. At certain region, the phelogen cut off closely arranged parenchymatous cell on the outer side instead of cork cell. This parenchymatous cell soon rupture the epidermis, forming a lens-shaped opening called lectancles. Lectancles permit the exchange of gases between outer atmosphere and internal tissue of stem. This occurs in mostly woody trees. 6.4.3 Secondary growth in roots. In dicot root, the vascular cambium is completely secondary in origin. It originated from the tissue located just below the phloem bundle, the portion of pericycle tissue above the protoxylem, forming a complete and continuous wavy ring which later become circular. Further events are similar to those already described above for a dicotyledonous stem. Thank you.